Welcome to the future of understanding. This is Neuromind Insights, where we unlock the power of your mind.
These rapid, aggressive pulses of neural firing across the brain's network characterize addiction, a clinical neurological disorder that physically targets the reward system. In a healthy state, this circuit performs a foundational biological task, reinforcing behaviors essential for survival, specifically feeding and reproduction. The brain maps these survival actions to distinct sensations of pleasure and reinforces them through memory formation. This internal loop ensures that whenever a physiological need arises, the organism is driven to seek out and repeat the behavior required to stay alive. Drugs of abuse hijack this exact evolutionary machinery, converting natural survival drives into a singular, overwhelming chemical dependency. This architecture begins in the midbrain, distributing signals upward into the limbic system and frontal cortex. It originates in the ventral tegmental area, or VTA. Triggers, like eating, cause the VTA to fire signals toward these limbic structures. These signals are carried by a specialized chemical messenger, the neurotransmitter dopamine. Beneficial survival activities stimulate the neurons in the VTA to manufacture and release this dopamine along the reward pathway. This release of dopamine from the VTA is the physical manifestation of what we experience psychologically as a reward. To see how this reward is processed, we must look past macroscopic structures and into the microscopic gap between two neurons, the synaptic cleft. The process is triggered by an electrical pulse, or action potential, that travels down the axon of the transmitting neuron to the nerve terminal. This pulse forces dopamine-containing vesicles to fuse with the cell membrane, dumping dopamine molecules into the synaptic space. The dopamine crosses the cleft and binds to specific receptor sites on the neighboring receiving neuron. This chemical docking is the specific event that generates the sensation of pleasure. For this system to be sustainable, the signal must be immediately terminated to prevent the receiving neuron from being dangerously overstimulated. This termination is handled by a specialized protein pump known as the dopamine transporter. These transporters vacuum dopamine molecules out of the cleft and recycle them back into the original transmitting cell. A healthy reward requires this balance: precise stimulation followed by rapid mechanical clearance. Drugs of abuse break this clearance cycle, causing the regulatory machinery to fail. One method, used by alcohol, heroin, and nicotine, is indirect excitation. These drugs force the VTA neurons to fire action potentials with abnormal frequency, flooding the cleft with more dopamine than the transporters can process. A second method is reuptake inhibition, the primary mechanism of stimulants like cocaine. Cocaine molecules lodge into the dopamine transporter proteins, physically plugging the pumps. Because clearance has stopped, dopamine is trapped in the cleft, where it repeatedly strikes the postsynaptic receptors. Methamphetamine employs a third, more invasive strategy: intracellular disruption. Like cocaine, methamphetamine blocks the surface transporters to prevent dopamine removal. It also enters the neuron, penetrating the membrane to invade the internal dopamine vesicles. This invasion forces the vesicles to dump their entire dopamine payload into the cleft, even without an electrical trigger. While the mechanisms differ, the pathology is identical: critical dopamine saturation in the synapse that causes prolonged, artificial euphoria. While the drug causes an acute flood, the brain responds with long-term biological defense mechanisms. Chronic overstimulation is toxic to the postsynaptic neuron. To survive these surges, the cell is forced to physically adapt. Through the process of desensitization, the receiving neuron systematically dissolves or retracts a large percentage of its dopamine receptors. Because of this missing architecture, the brain now has a blunted capacity to process everyday stimuli. Natural rewards, like food or social interaction, still release dopamine, but the amount is too small to activate the heavily depleted receptor pool. This physical deletion of receptors shifts the user's priorities. The environmental baseline is destroyed, leaving the drug as the only chemical powerful enough to trigger the system. Physical drug tolerance is the mechanical result of this receptor downregulation. To overcome this high reward threshold, the user is forced to consume escalating doses of the drug. This creates an accelerating failure loop. Higher doses lead to further receptor loss, which in turn requires even higher doses. Eventually, the receptor pool is so depleted that even extreme doses fail to produce a reward. This chart shows receptor density collapsing while the required dosage spikes. As the user chases a vanishing high, the drug concentration reaches levels that overwhelm the body's other physiological systems, such as cardiac rhythm or respiratory drive. A drug overdose is the mechanical failure of a biological system pushed far beyond its evolutionary parameters by chemical hijack.
Thank you for watching. If you enjoyed this video, please like, comment, share, and subscribe to Neuromind Insights for more.